Welcome back to Immunology course. Today we are going to start again the basic concepts in immunology. Today is lecture 4. Okay. So, in last class we ended up with the dendritic cell is the bridge between innate and adaptive immunity. Today what we are going to see that how that innate immunity and adaptive immunity are bridged together and what is happening and how it is going. Okay. So, the first slide we are talking about circulating lymphocytes encounter antigen in peripheral lymphoid tissue. If you see the picture carefully, see this is the place in the leg, okay, in the foot actually the infected peripheral tissue. So, there are some infection here. Okay. So, what happened in infection, if you remember, in infection the first thing happened is the innate part of the immunity and this is the inflammatory response. In inflammatory response we already discussed. So, all these macrophage dendritic cells they pick up the tissue or pick up the infected organism, infectious organism, and then it bring to the nearest lymph node. Okay. So, from the site of infection the antigen presenting cells mostly the dendritic cells are taking them to the nearest lymph node, and we already know that lymph node is spreaded all over the body and wherever the infection. Here in this case we show that the foot is the site of infection. It can happen any part. So, that from innate as a result of innate immunity the infectious organism is processed and brought to the nearest lymph node region. In this case it is here. Okay. So, in this lymph node it will go and we will discuss what is going to happen in the lymph node, and then it will go to the circulatory system, goes to heart, and from that heart it is circulated all over the body. If the site of infection is only one place, it will take care there, and if it is all over the body, then it will take care by the blood. So, like after inflammation and the dendritic cells bring them to the nearest lymph node. So, how the lymph node look like? This is a cross section of lymph node, the schematic diagram. Okay. So, if this is the lymph node and here there are two channels, one it is called afferent lymphatic vessels where it is coming from outside entering into the lymph node, and other is going out after the immune reaction happen. This is called efferent lymphatic vessels. In this lymph node cross section, if you see there are different parts. This part, the pink color part, it is the medullary part of the lymph node, and this region is cortical region. Okay. If you remember the color code, that we always show that B cell is yellow in color and T cell is blue in color. Okay. So, here this blue and yellow are meaning the same part, like blue region means this region is packed with T cells. Okay. And this color, this pink region is medullary cord, macrophage and plasma cells are there. What is plasma cells? We will discuss later, but we already told actually like B cell is converted to plasma cell and then only it can secrete, or the plasma cell is actually producing the antibody molecule and secreting. So, here this yellow circles, this is called germinal center. Okay. So, what happened in previous slide we have seen that the dendritic cells are bringing the pathogen here and then it is entering inside. Then in the blue region there are lots of T cells, and these T cells, this we already discussed this slide, these T cells there are many. Okay. The specific T cells, specific or particularly interacting T cells which interact with that pathogen will find it and attach to it. After attachment they get the signal and they proliferate. Proliferate means they increase their number. Okay. These activated T cells then help the B cells to produce antibody, and B cell also after getting the signal it multiply converted to plasma cells, and plasma cells produce antibody which is secreted to the system, and peripheral system that goes to heart and then gradually distributed all over the body. So, wherever the infection will find, the antibody is going to do its own job. Okay. So, not only lymph node, there are two types of lymphatic organ, one is called primary, another is secondary. So, another important lymphatic organ is spleen. Okay. So, spleen also, if you see the cross section of spleen, here also you will find two different zone, one is blue zone, another is yellow zone. So, blue zone is full of T cells and yellow zone is full of B cells. Okay. So, there are different part. So, the germinal center is also here. So, it depends where infection happen, which is the nearest lymph node or lymphatic organs. Say for example, if you go here. So, if there is any infection in teeth or mouth or in throat, we are very much acquainted with that. What happened? We heard like tonsil swells, right. Tonsil is located here. So, tonsil is taking care of all this region, infection in this region, in the mouth cavity, throat. So, any infection happen. So, that time the macrophage and dendritic cells bring the infection to the tonsils. Okay. So, similarly, it is taken care by the nearest lymph node, which is I called in the last class or in last one of the last lecture, that it is the local khana kind of thing. So, it is taking care immediate part. Okay. So, lymph node, spleen, tonsils are there. So, besides this, in case of the mucosal region where there is no lymph node or there is no direct connection of blood. So, what we also have several system or several mechanism to handle the infection in that cases. Like mucosal surface have specialized immune structure which take care of the infection of that region. What we have? We have mucosal immune system or mucosa associated lymphoid tissue. It is called MALT, or gut associated lymphoid tissue GALT, which includes tonsil, adenoids, appendix, Peyer's patches, nasal associated lymphoid tissue and bronchus associated lymphoid tissue. In short, we call MALT, GALT, NALT or BALT. Okay. So, they are taken care by a specialized cell called M cells. Okay. So, this is a specific example of the Peyer's patch, but Peyer's patches are covered by epithelial layer. Okay. You can see the epithelial layers are there, which contain a specialized cell called M cells, which have the characteristic of the membrane ruffles. So, these ruffles are there. So, here also we can see there are packed of T cell region, the blue region, we have germinal center, dendritic cells are migrating in between. Okay. So, there they take care of the infection if there is any in the mucosal region. All the say GALT, MALT, everywhere they have this kind of structure. So, what happened? I am revising back or repeating the same thing. What happened? In the site of infection, immediately the innate immunity take care. Inflammation happened. That inflammation, as a result of that inflammation, the pathogenic organism brought to nearest lymph node, where it interacts with T cells. T cells get activated, it helps B cell also activated both way directly by the antigen and at the same time by helper T cells, then they convert it to plasma cell, produce antibody. Okay. In many times it happen, like if you go to doctor, what happened? Doctor first, like there is a pain or there is wound, something happened, muscle pain, doctor check whether the gland is swelled or not, because inflammation will make the gland will swell if there is a good amount of infection, and there will be a pain in the lymph node also or lymph node region. So, from that pain, if pain is there as well as there are some wound, we can say inflammation happen. So, that means infection is there. If there is no swelling, just pain happen by some heat to the muscle or by some overwork, that time inflammation is not going to happen. So, from there doctors can understand infection is there, and according to their prediction and other symptoms, they decided what should be there for the treatment, whether antibiotic is required or antibiotic is not required, they will decide. Okay. So, this is the starting of adaptive immune response. So, if we consider at the beginning just the B cell part or antibody production part, what happened? So, it is not like innate immunity within hours, we already see that it takes days to week. What happened? So, first antigen will come to lymph node, B cells, there are many B cells, they will see specific B cell with their receptor will recognize the antigen. Same way specific T cell receptor will recognize, between there will be a crosstalk between B cell and T cell as well as with the antigen, that will activate the B cell, which is going to convert it to plasma cell, and the B cell receptor will be produced in a different form because there will be no transmembrane domain. So, it will be now secretory. So, the B cell receptor in the form of antibody, same molecule, same specificity will secrete to the peripheral system. So, here we are showing a graph in this picture. This is a very important in that sense. This is the response, how it goes, and this particular graph is also going to, I am going to tell you that this is the major principle, or on this principle or this phenomena what we are going to explain now are the base of immunization or the basis of immunization. So, I am trying to explain this graph. What is happening? Say for antigen A, okay. Suppose we are considering antigen A, and assume for the time being, we will discuss later how, assume for the time being in blood serum we can measure how much specific antibody is there. So, antibody against antigen A we can measure, there are several techniques. So, suppose one of the technique is used to measure the specific antibody concentration in blood against antigen A. So, what happened? Suppose antigen A is injected, this is day 0, okay. And every day if you try to measure, or if you measure the specific antibody, presence of specific antibody in serum, what we will see? Up to almost 7 days there is no increase in antibody against antigen A, and after that it gradually goes up, okay, and it reach a plateau. That means, antibody is not going to increase anymore. We injected antigen A once at day 0. So, up to certain day it will increase, and then it will not increase anymore, and if we keep on continuing the measurement of antibody, what we will see, it is gradually going down, okay, going down. At this moment, suppose here we are injecting antigen A again, listen carefully. We first injected antigen A, then we measure the antigen A specific antibody in serum, and we see the pattern of this graph. So, first 7 days almost there is no increment, then it is increasing, then reach maximum, stay for some days, and then going down gradually. When it is going down, at this moment if we inject the antigen A again, what is happening? You see, this is the nature of graph. Nature of graph means this is the nature of antibody concentration in that particular individual. So, what is happening? If you see, in the beginning it took almost 7 days. Okay. Next time if I inject same antigen, within very short, maybe 1 or 2 days, it start increasing. Not only it takes less time to produce antibody, it produce much more, okay. And if you see, it stay for long time, okay. This is called secondary response. So, first one, when our body see any antigen or any pathogen first time, this is the first reaction, but second times if it sees the same antigen again, the reaction is much quicker and the amount of antibody production is very high. So, two different. So, if I now ask you a question, what is the difference between primary response and secondary immune response? There are 2 points, right. One, the lag phase, that means first point where it goes up, the lag phase is more, it is about 7 days, here it is much less, 2 to 3 days. Okay. This is number 1. Number 2, the amount of antibody production in the primary immune response is much less than the secondary response. The amount is very high and this is independent of antigen. Okay. It is not that antigen A, if happen this way, it is going to work for antigen B also. That is why here what happened, along in the second time injection of antigen A, antigen B also injected. You see, in case of antigen B, again it took 7 days, okay, to start with, and again it goes up to certain level. So, for every antigen, first administration is the primary response. So, secondary response of antigen A is not doing any effect or not having any effect for antigen B. And this is the basis or this is the principle of immunization. What we are doing in immunization? We are doing immunization, we are giving our body to the exposure of the antigen for primary response. So, in primary response, body see it, it takes time, make a specific antibody. So, what is going to happen when it is going down? It is never going to come down to the basal level. Some of the B cell will convert to memory cell. That means that it will remember that okay, who entered in our body. So, that one is that population of B cell, the memory B cell and memory T cell are going to stay in our immune system. So, next time if it comes, it will not take much time, because they already know it is how it looks, what to do with that. So, immediately they will take care of that, and with much less time they goes up and make more production. Okay. There are 2 more important thing happen, one I will say now, another I will come later during the discussion of antibody. What happened? This secondary immune response, it is not only that it is quick and much more aggressive, another thing is happening. In the secondary response, the quality of antibody or the specificity of the antibody is increased. So, better quality antibody or more specific antibody are going to found. So, in secondary response, it is always better. It is always better. So, that is how vaccination or immunization work. So, we prime our body with the antigen at the first time, and in secondary response it goes high and can take care of our infection much quicker and much specific way or more effective way. So, what is the effector mechanism? Effector mechanism is what will happen, what this antibody is going to do, because antibody is produced. So, more antibody is produced. So, how it is going to handle the immune system? We already know that there are 4 type of pathogens, right. The pathogens are like virus, which is intracellular, then intracellular bacteria or protozoa like Listeria is intracellular mycobacterium, Mycobacterium leprae, Mycobacterium tuberculosis, they are intracellular bacteria. There are some protozoan parasite like Leishmania donovani, which cause Kala-Azar, which grow inside the macrophage. Plasmodium falciparum, you know, it grow inside the RBC. Similarly, which is very common, the extracellular bacteria, parasites, fungi. Okay. Any kind of tissue infection is most of the time it is extracellular. There is Streptococcus, Clostridium, Trypanosome is a protozoan parasite, then parasitic worm, the big worm, Schistosomiasis, Ascaris, and here is the disease, the viral disease like smallpox, flu, chicken pox, then leprosy, leishmaniasis, malaria, toxoplasmosis, they are the disease, this is the corresponding pathogens. So, summary, if I say summarize this, we do not have to remember all these things. If you remember, it is good, like which organism cause which disease, but in general we have to remember there are 4 types. One is virus, pathogen that antibody can handle. Pathogen is the virus, intracellular bacteria, protozoa or parasite, extracellular bacteria, parasites, fungi, and parasitic worm which is also extracellular. So, immune system or the effector function of antibody is going to handle all these 4 different kind of organism or pathogen. How they are doing? If you go back, so virus is going to take care by cytotoxicity of the cell, cytotoxicity mostly by NK cells and CD8 T cells. Okay. I will come later, but for these slides cytotoxic T cell also called CD8 T cells, because cytotoxic T cells express a specific receptor which is CD8, which is present only in cytotoxic T cell. Okay. Similarly, helper T cell produce another receptor which is specific to helper cells are CD4. So, many times instead of telling helper T cell, we also say CD4 cells. Same way cytotoxic T cell also we call CD8 T cells. CD4 T cells are the helper cells, CD4 T cells are the helper cells, CD8 T cells are the cytotoxic cells. So, virus is mostly taken care by NK cells, which we already discussed, by and cytotoxic T cells or CD8 T cells by cytotoxicity. What it is doing? Elimination of virus infected cell or metabolically stressed cells. Intracellular bacteria or parasite, ILC1, innate leukocyte 1 or Th1 cells, we will see what is that, which eliminate the intracellular pathogen by activating macrophages. Similarly, mucosal barrier is taken care by ILC2 and Th2. The effector function is elimination and expulsion of parasites, recruitment of eosinophils, basophils and mast cells to handle the infection. Extracellular immunity is taken care by ILC3 and Th17 cells. What they are doing? They are eliminating the extracellular bacteria, fungi and recruitment and activation of neutrophils, which is taking care of the innate immunity part. Okay. So, now what antibody is doing? All these thing is going to activate B cell, T cell, and B cell T cell is going to help B cell, they produce antibody. How antibody is going to help us to get rid of the infection or infection related material, because some extracellular bacteria they produce toxins and that toxin create problems like diphtheria toxin, cholera toxin, okay, tetanus toxin, then pertussis, the whooping cough, okay, all are not direct bacterial is causing anything. So, they are producing a protein molecular toxin molecules. What they are doing? So, they release the toxin molecules, these red circles, the red circles are the toxins. Normally, what they are doing? These toxin molecules we are attached or bind to receptor which is already present in our cells. So, bacteria tamer produce the toxin to kill us. Our cell actually have the receptor which can bind these toxin molecules. Okay. So, that is a very interesting mechanism. Those who are interested how these toxins work, you can go and read like diphtheria toxins, how they go enter and change or attack the elongation factor and stop protein synthesis. Cholera toxin change the sodium potassium channel and a lot of fluid loss is happening. And tetanus toxin cause the problem in muscle construction. So, their mechanism or molecular mechanism are very interesting. You can go and read, study, not for immunology, just for general interest. What happened? So, these toxin molecules go and bind, they internalized by receptor mediated endocytosis and cause the damage or disease. Okay. So, if there is any specific antibody which raised against this toxin, what is going to happen? This toxin is not going to be free anymore. Okay. The antibody is going to bind them. If you see this antibody is just captured them. Okay. So, what will happen? There will be no free toxin to bind to the receptor, and this particular toxin is captured by the antibody. So, upon infection, body is going to make specific antibody against this toxin, and when antibodies amount is good enough in our body, so no free toxin will be there to bind to cell receptor and cause the damage. Okay. Same way, this is called neutralization, this method. So, it is, that means the toxin is neutralized by antibody. This process is called neutralization. Same way, macrophage, we know that they can eat bacteria by different TLRs, okay, managed receptors, but sometimes what happens, that efficiency is not good. Even after that innate immunity, if bacteria grow inside our body and population increase, so the disease will continue. So, what happens, if it is still continuing for good enough days like 7 days or more, then antibody against this bacteria will be produced. So, what will happen, just like the toxin molecule, antibody is going to coat the bacteria. Okay. So, the bacteria is big here, and so these bacteria will be coated by antibody. Okay. This process is called opsonization. Clear? So, first one is neutralization, second one is opsonization. Another important method, strategy is there in our immune system, which is called complement activation. What is this complement activation? There are proteins which is heat labile, but they are very much part of the innate immunity, they do not have specificity. What is going to happen, as soon as bacteria is coated by antibody, the complement protein which is present in our blood, okay, they will go and attack them. This is called complement activation. So, antibody is doing three things: first neutralization, then opsonization, then complement activation. Complement will be taught separately. There is a very interesting mechanism how it works, how our own cells are not affected by this complement, okay. But they are very, very bad proteins. Okay. So, human complement can kill any other mammal's cell. Okay. So, they are very specific. Our own complement is not going to do anything to us. Similarly, others complement system, like if it is cow or goat, any complement system, if you mix with our blood, that will kill our own cells. So, they are non-specific, highly non-specific, but individual organism had the protection from their own complement system. Okay. So, coming to the previous slide here, neutralization, opsonization and complement activation. I will go one by one. So, after neutralization of the toxin molecule, what happened? Macrophage is there. Macrophage, I already told that it is cleaning mess or eating our own thing is there. Macrophage has, if you see this yellow line, macrophage has a receptor for antibody. Okay. So, they by this receptor, this whole complex will attach to the macrophage surface and they will taken up by phagocytosis or receptor mediated endocytosis. After taking them, they will be degraded inside. So, there will be no toxin molecule anymore, okay. And this whole protein is going to be recycled for our own, I mean the macrophage own protein synthesis. Same way, opsonized bacteria which is coated with antibody also be endocytosed or phagocytosed by macrophage and degraded inside the macrophage. So, no bacteria will survive. So, that is the way the bacteria will be cleared from the system. So, this is opsonization, and end product is same, both are lysed product of the antibody and toxin. In complement activation, what happened? In complement, as soon as the antibody coat this bacteria, complement protein come and sit into this bacteria. Okay. So, they will make a channel. Okay. There are proteins which are like this. So, this suppose this is one protein, one subunit of the protein. So, one subunit, then two subunit, three subunit, and then gradually they make a channel. It is just like a pipe. Okay. So, if you take a pipe and put inside the cell, what I mean, say suppose a ball is full of liquid, you put a pipe inside it, what will happen? All the liquid from out cell will from that ball will come out. Same way, so they make a channel inside that. Okay, so individual protein will sit one by one. Okay, and how it is happening? How it is happening? So, they will make one protein, then 2 protein, then 3, 4, 5, 6, 7, something like that. So, they will make a channel or hole in it. Okay. So, they will make a hole in it. So, that hole, what is the effect of that hole? So, all the cytoplasmic material of the cell will come out. So, the bacteria will lyse. So, these 3 possible way are there. Okay. So, these 3 possible way are there by which this antibody can take care of microbial infection, either the toxin or the whole microbe. Okay. So, this is called effector function. Is that clear? Okay. So, today I mean in this lecture, this is the end. So, see you in the next class, and you just again I am repeating, go and study. It is very simple and things to remember. Just go, and if you remember the image, you can remember the whole immunology. Bye for